Hello everyone, welcome to my channel. Today I'm going to talk about different organelles in part 5, and in case if you have missed 1, 2, 3 and 4, please check them out. So I'm going to teach by following NCERT textbook from page number 137 today. So I'll be covering cilia, flagella, centrome, nucleus and microbodies for today. All right, so let's get started. And in case you did not subscribe to my channel yet, please do subscribe so that you will receive a notification, and you can always leave the topic of your interest in the comments below and I'll try to make a video on it. So coming to cilia and flagella. So cilia and flagella are hair-like outgrowths of the cell membrane. They're nothing but hair-like outgrowths of cell membrane. All right, flagellum is something like this. Okay, so you must have seen in paramecium how cilia will be there, right? They look something like this. Okay, so cilia are small structures which work like oars, and what is the main function of them is they cause the movement. Okay, flagella are comparatively longer and they are also responsible for cell movement. Okay, remember one thing, cilia and flagellum both are covered with plasma membrane. All right, now this structure over here is called as 9+2 array. And why do we call this? We'll be talking about that soon. Okay. So the electron microscope study of the cilium or flagellum show that they are covered by the plasma membrane. All right. And their core is called axoneme. And they have lot of microtubules. All right. So what is their core called? Their core is called as axoneme, and they have lot of microtubules. And if you see the picture, you can see that they are parallel to the long axis. Okay. Now, the axoneme usually has 9 pairs of doublets. Now, you might be confused, like, where do I see 9 pairs? So this is pair 1, pair 2, pair 3, pair 4, pair 5, pair 6, pair 7, pair 8, pair 9, right? And you have 1 and 2. So that's why you have 9+2 arrangement. So you have central microtubules, there are 2 in number, and you have peripheral microtubules, 9 in number. So let us write over here. So how many peripheral microtubules do we have? We have 9. And how many central microtubules do we have? We have 2. All right. Now, so if you see this attachment, like you can see how these microtubules are having interdoublet bridges over here. Okay, here you can see them. And then you have, so this is the radial spike over here. All right, so I'm just drawing it again so that it's easy for you. So these are the radial spikes. These are the interdoublet bridges, which are like basically attaching two doublets, like here, which you can see the small ones, they are interdoublet bridges. Okay, then you have a central sheet, and you can of course see the outer plasma membrane. Yeah. So I hope we are clear till now. So what did I explain till here? That cilia and flagella are like hair-like outgrowths, all right, of the cell membrane. And cilia are small structures, whereas flagellum is a longer one. And both of them help in cell movement. Okay, and they're covered by plasma membrane. and they have 9+2 array. They have 9 peripheral microtubules and they have 2 central microtubules. And like if you see, each of the pair of the peripheral microtubules are attached to each other with the help of interdoublet bridges, and they have radial spikes on each of the pair. And here inside the central sheath, you can see 2 central microtubules. Okay. Now, let us talk about nucleus. But before we talk about nucleus, let us talk about centrosomes and centrioles. So centrosomes look like this. Okay. So inside the centrosome, you have centrioles. So if this is the centrosome, this is the centriole. All right. Inside the centrosome, you have centrioles. So what is a centrosome? It is nothing but an organelle containing 2 cylindrical structures, and they are called as centrioles. They are surrounded by pericentriolar membranes. Okay, so if you have to if it's asked as like a 2-marker or 5-marker to talk about this organelle, what can you say is it is an organelle containing 2 non-membrane bound cylindrical structures. So it is 2, is it membrane bound or non-membrane bound? It is 2 non-membrane bound cylindrical structures, all right, called centrioles. Okay, and they are surrounded by pericentriolar materials, and they always lie perpendicular to each other. Okay, and they are made up of, this is very important, they are made up of peripheral fibrils of tubulin. They're made up of peripheral fibrils of tubulin. All right, so these are the things that you need to remember. So centrosome is an organelle which is containing 2 cylindrical structures, okay, and they're always perpendicular to each other, and they are non-membrane bound, and they're made up of peripheral fibrils of tubulin. Okay, so if they ask you like, is centriole inside the centrosome or centrosome inside the centriole, always remember that centriole is inside the centrosome. Okay, now let us talk about nucleus, which is very important. Now, nucleus is of course a membrane-bound organelle that is called nuclear membrane, like here you can see, right? So it is a membrane-bound organelle. All right. Now, endoplasmic reticulum is attached with the membrane. Remember that. Okay. Now, what is present inside the nucleoplasm? So the matrix inside the nucleus is called as nucleoplasm. Okay. And the type of solution present inside it is a colloidal solution. All right. And most important NEET question in this particular slide over here is, where is the synthesis of rRNA happening, ribonucleic acid? rRNA happening. It happens in the nucleolus. So remember, rRNA synthesis happens in the nucleolus. Okay. Now, coming to chromosomes. Chromosomes are nothing but DNA's in packed forms, okay? So this is all DNA in its packed forms, okay? Every chromosome has a centromere, and this is a primary constriction, okay? So centromere is a primary constriction. And every chromosome has kinetophores, okay? They are like disc shaped, all right? So based on the position of the centrosomes, there are 4 types of chromosomes, okay? So here you can see that centromere is very much in the center, Here you can see that centromere is not in the center, like there is short arms and then long arms. It's not equal arms, right? And then here you can see centromere is too much at one end, right? And here you can see like it's completely at the top. Okay. So based on that, so how did we divide these chromosomes? Chromosomes are divided based on position of centromere. All right. So they are divided based on the centromere. So if centromere is in the center, then it is called as a metacentric chromosome. And if centrosome is little away from the center, it is called as submetacentric chromosome. And if the centromere is like very much far, then it is called as acrocentric chromosome. And if like you can't see one side end because it's completely, the centromere is at the top, then it is called telocentric chromosome, which means it only has a terminal centromere. All right. So here in the metacentric chromosome, it has middle centromere, right? That's why the arms are in equal length. Now coming to submetacentric. So this is submetacentric and this is acrocentric. So in case, let me explain. So here you can see centrosome is exactly in the center, right? So it is called as metacentric chromosome. Okay, so it has a middle centromere, right? Therefore, the arms are equal. Now we have submetacentric chromosome. Okay, submetacentric chromosome means the centromere is not in the center. Okay. So what happens? This is the short arms and this is the longer arms. Now, in case of acrocentric chromosome, centromere is situated close to one end, like all right. So to make it see this one, this is called acrocentric chromosome. All right. Then we have terminal centromere in case of telocentric chromosome. Okay. And what happens during anaphase? Segregation happens, right? Based on the segregation that happens during anaphase. In case you don't know what is anaphase, I can make a video on that, but please leave in the comments below because this is covered in your earlier classes, so in your high school. So still, if you want me to explain in detail, I'll be explaining, or else I'll be covering it in the class 12 topics. All right. So based on the segregation during anaphase, chromosome shape is, so how do you think the shape of the chromosome over here is? This is like V-shaped. This looks like L-shaped. All right. This becomes J-shaped and telocentric becomes I-shaped. All right. Now here there is something called a satellite. So some chromosomes have a secondary constriction, all right, at a constant location. That is called satellite. So what is satellite? Satellite is nothing but chromosomes having secondary constrictions at constant location. Okay, that is called as satellites. Now, coming to microbodies. So microbodies, you can see here a lipid bilayer and it is surrounded by plasma membrane and you have a crystalline core. Okay, they are of course membrane bound, as you can see from the picture. They are nothing but membrane bound vesicles. Okay, they contain various enzymes and they can be seen in both plant cells and animal cells. Okay, so this was the last organelle And this is the end of your page number 140, and the lecture cell ends over here. I can do the questions that are there in your NCRT as well as NEET questions that come from cell topic if you're interested. All right, so let's have a quick revision of what we have studied today. All right, so I'll just change the ink color for the revision. Okay, so cilia and flagella. Cilia is more like they are thin and then they are more in number. Flagella is like long, and both of them help in cell movement, and they are covered by plasma membrane. They have this 9+2 array, which means they have 9 peripheral microtubules and they have 2 central microtubules, and these 2 peripheral microtubules adjacent to each other are connected with the help of interdoublet bridges, and each of this microtubule has a radial spike, and you can see in center there are 2 central microtubules. That's why it is called 9+2 arrangement. Okay, now coming to centrosomes. Centrosomes are nothing but they are non-membrane bound organelles, and we can see centrioles inside the centrosomes. All right, and they are perpendicular to each other, and they are made up of peripheral fibrils of tubulin. Nucleus is of course surrounded by a membrane. It is called as nuclear membrane, and endoplasmic reticulum is attached to this membrane. And in the nucleoplasm, the type of solution you can see is a colloidal solution, and the RNA synthesis, rRNA synthesis happens inside the nucleolus. So this is the nucleus, this is the nuclear membrane, and this is nucleolus where rRNA synthesis happens. All right, then you can see the nuclear pores. Now, DNA is packed in the form of chromosomes, and based on the position of each chromosome has a centromere and kinetophores. Based on the position of the centromeres, the chromosomes are divided into 4 types. One is metacentric chromosomes, then you have submetacentric chromosomes, you have acrocentric chromosomes, and you have telocentric chromosomes. All right. Here, centromere is exactly in the center, and in submetacentric, it is a little bit away from the center. Acrocentric, it is very much away, that the small arm is really, really small. And in telocentric, like you can see that centromere is in its terminal position. And satellite here, what you can see on chromosomes is nothing but some of the chromosomes have secondary constrictions at constant location, and it is called as satellite. Microbodies. Microbodies are nothing but membrane-bound vesicles, and they have various enzymes, and they can be seen in both plant and animal cells. Here you can see the plasma membrane very easily. So of course it's a membrane-bound organelle. All right. So thanks for watching my channel, and stay tuned to my channel for more videos. Thank you. And in case you require any notes on NEET topics, please let me know so that I can provide you guys with the PDF link. Thanks.